Thank you. It's such a pleasure to be here and talking with you. Sangamo has 3 late-phase products. It has hemophilia A that's partnered with Pfizer that is in phase 3. It has Fabry, which is a wholly owned product that's about to go into a phase 3 with Sangamo. And we have our sickle cell asset, again, wholly owned by Sangamo, and again, just about to go into phase 3. And it's so exciting for a company like us to have 3 late-phase assets. But behind that, we have an incredible pipeline. We have dosed the first ever CAR-Treg patient, really exciting, in renal transplant with a whole portfolio of those to follow. And then something that we're excited to talk about at this meeting is our CNS pipeline. And that's the most incredible thing about Sangamo. As well as having clinical programs that are making a big difference to patients, already we see the benefit to patients. We have decided to invest in our science. We have a marvelous platform, and we have a great bunch of scientists led by Jason Fontenot. At this conference, there are 8 abstracts, and what appeals to me about them is the breadth and the depth. Each one is cutting edge, leading the field for their subject. But the breadth of them is incredible. We have a new base editor that we feel has the potential to be best in class. We have capsid engineering that will evolve the capsid to be able to deliver to the CNS. We have transcription factors for T cells, which will get rid of that double-stranded break issue and allow a much easier way to create CAR-T. And we have B-cell engineering. And the breadth of that is incredible from a company our size. Genomic medicines, the challenge is delivery. It's all about delivery. Our technology works. In the cells, zinc fingers will work every time and will give you the editing, the repression, the base editing, the integration that you need. It's all about delivery. And at the moment where we are hampered that the best AAVs take you to the liver, which is fine for rare diseases in the liver. But the future, I believe, is CNS delivery. And with the current viruses, it may be possible to deliver via the cerebrospinal fluid. But as a patient, that's not ideal. You would much rather have something that could be delivered intravenously. And so at Sangamo, we have 2 very bright young scientists, David and Matt, who are looking at capsid evolution to both find the best capsid to deliver via the CSF or intravenously. And it's remarkable what they do with barcodes and gazillions of viruses. They gradually evolve them till they get to the brain. And once we get there, we'll be able to address diseases like Alzheimer's, Parkinson's disease, Huntington's disease, prion disease, and be able to address such high unmet medical need. So Sangamo's got this wonderful platform. It's been developed from zinc fingers, which are one of the commonest things to control your genes in the body. So it's a eukaryotic, natural human version of gene control. It's small, it's about 1/10 of the size of CRISPR, and that allows it to both be packaged easily and also to get in underneath the chromatin and to be able to, we can lay down zinc fingers anywhere in the genome without restriction or PAM sequence requirements. But I think of it as a Swiss Army knife. I think of the zinc finger, the bit that takes you to the DNA, as the red bit of the Swiss Army knife. And then you've got all these pieces that you can open out from the zinc finger localization. You can use nucleases, you can use transcription factors to repress, like we do for Biogen, to enhance, like we do for Novartis. We can add a base editor, we can do epigenetic engineering, we can even think about integrases. And that's why we've been so successful in our partnerships, because there are now 27 CRISPR companies. Each one does a little different thing. And you go to each one for a different tool, or you come to Sangamo and you get the toolbox that has everything in it that you need. And that's why the pharma companies come to Sangamo and love working with us, because the science, you fit the science to the disease, not the disease to the science. Oh, what a good question. With all scientific discoveries, there is a moment where everyone thinks it's the best thing since sliced bread, it will solve everything. And then a moment of reality where they despair that it isn't as good as it was thought. And then a gradual evolution of understanding of which patients it does the best in and in what regimes it works best in. So the CAR T are mainly CD19 at the moment, and they have great effects there, but we need to better understand which patients we should target against. I think the evolution of CAR T is understanding beyond the blood tumors into the solid tumors, and that I think is more challenging. But if we're successful with that and successful with creating allogeneic forms of that could be given off the shelf to the patient urgently when they appear, I think that we will, as with all oncological treatments, gradually push back cancer and eventually make cancer a chronic disease of living.